Today's topic is non-targeted analysis and suspect screening, where we will talk about the databases, tools, and application examples. In this lecture, you will learn the definitions of targeted, non-targeted, and suspect screening. The non-targeted analysis workflow and the common mass database for non-targeted analysis and the applications. So the targeted analysis in mass spectrometry refers to the intentional and specific analysis of selected molecules or compounds in a sample. It is performed by selecting specific precursors, fragments, or reaction products and measuring their mass to charge ratio to identify and quantify the target compounds. Suspect screening in mass spectrometry refers to the process of identifying unknown compounds in the sample by comparing the mass spectral data of the sample with the reference database. This approach is used when the specific identity of a compound is not known and a general screening of the sample is needed to identify potential target compounds. In suspect screening, the mass spectral data of the sample is compared to a reference database, such as the National Institute of Standards and Technology (NIST) library, to identify compounds that have a similar mass spectrum. This process can be performed using the software tools that perform database searches and provides a list of potential matches along with their confidence score. Suspect screening can be performed in a targeted or untargeted manner. In targeted suspect screening, the search is limited to specific classes of compounds, such as drugs or toxins, whereas in untargeted suspect screening, the entire database is searched for potential matches. Non-targeted analysis is mass spectrometry, refers to the process of analyzing a sample without prior knowledge of a specific compounds that are present. Unlike targeted analysis, where specific compounds are selected for analysis, non-targeted analysis aims to identify all compounds present in the sample, regardless of whether they are known or not. The term non-targeted analysis (NTA) now is commonly used to describe both suspect screening and untargeted experiments. These slides summarize the brief definitions of each type of mass analysis. Targeted analysis is with standards and a calibration curve for all analyzed. Semi-targeted analysis refers to semi-quantitative analysis of many analyzed with a calibration curve of one representative standard. Suspect screening does not require any standard but need a mass database. Untargeted analysis requires no standard and no mass databases. In the context of food safety, food frauds can be put into three categories: origin, substitution with cheaper similar ingredients, or extension of food. This highlights the large variation in food authenticity issues and indicates that evaluations of a single analytical parameter, either chemical or biological, is often insufficient to reliably verify the authenticity of a product across these categories and different food commodities. More recently, advanced analytical approaches such as profiling and fingerprinting, accompanied by multivariate statistics or chemometrics, have shown potential in this respect. This figure graphically presents the principles of targeted versus non-targeted authentication together with a conceptual presentation of primary versus secondary markers. The first horizontal arrows from the left illustrate if the single or dual target, several targets, unspecified targets or data points are measured. Red, blue bullseyes represents the primary marker. Yellow bullseyes represents a secondary marker. And a gray dartboard illustrates that no specified targets are addressed, but that the major targets or data points are still within a closed entity, such as proteomics, metabolomics, etc. The arrow after the dartboards illustrate the fate of the analytical results, either a direct authentication result or an indirect authentication result following a comparison to a legal limit, a threshold value or database or reference. The indirect authentication procedures often include the use of a multivariate statistical analysis. Here are some basic definitions for the terms used in the previous slide, just for your reference. In the following slides, we will go through the typical workflows for non-targeted analysis. These slides illustrate the typical methodological pipeline of an untargeted metabolomic study. This methodological pipeline starts with the processing of spectral data to generate the sample metabolic information or the metabolic features, together with the metabolite identification method. Spectral processing methods are highly dependent on the analytical techniques used, like NMR, LC-mass, or GC-mass. Once the complete set of metabolomic features has been generated, univariate and multivariate data analysis method can be applied to investigate the general structure of the metabolomic data in the dataset and how the different metabolic features are related with the phenotype data associated with the sample. The analysis of metabolomic data can often be used to build models that attempt to describe the observed data and to have a high-tier biological interpretation. Spectral processing is a methodological approach aimed to accurately identifying and quantifying the features in the sample spectra of a metabolomic study. Metabolomic spectra are sequentially or jointly processed until a final set of feature quantification is obtained. Spectral processing is also necessary to guarantee that each final measurement will refer to the same metabolomic feature in all samples. The data resulting from the spectral processing is generally arranged in a feature quantification matrix that contains the quantification of the metabolic features of all the analyzed samples, and that will be used as input for subsequent statistical analysis. In order to improve the signal quality and reduce potential biases present in the raw data, several pre-processing steps are usually applied. In NMR and MassBank spectra, baseline correction is used to remove low-frequency artifacts and differences between the samples that are generated by experimental and instrumental variation. After this, the application of high-frequency filters may be necessary to remove the electronic noise present in the data that is generated by the measurement equipment. The objective of the feature detection step is to identify and quantify the features present in the spectra. Peak-based methods are the most common algorithm choice for feature detection in mass-based studies. These methods detect the peaks across the spectrum and integrate their areas to provide a quantification of the underlying metabolite. Peak detection includes two analytical steps: smoothing and peak filtering. In the first step, the spectra are smoothed using different filters such as a moving average, Gaussian distribution, etc. In the second step, the different metabolite peaks are identified using one or multiple detection thresholds. These thresholds are applied to different parameters such as the signal-to-noise ratio, the intensity, or the area of each peak from the resulting filter spectra. In metabolomic studies involving large number of samples, a frequency filter or constant peak signal can be also applied so that only those peaks that are present in the maximum percentage of samples in the same group are selected for downstream analysis. Spectral alignment is one of the main processing steps in metabolomic studies involving multiple samples. When analyzing multiple spectra, the position of the peaks corresponding to the same metabolic features may affect by nonlinear shift. In mass-based studies, peak shifts are mainly observed across the retention time axis and are generally associated with changes in the stationary phase of the chromatography column. Spectral alignment methods must be therefore applied to correct these undesired variability in the samples that can profoundly affect the quality of the study. Spectral alignment algorithms can be divided into two main groups: spectral alignment methods, where the spectral data is aligned before peak detection, and peak-based alignment methods, where spectral peaks are aligned across samples once they have been detected using their coordinates. We will introduce a useful spectral processing tool for spectral alignment, Skyline Mass, in the later slides. In order to perform an accurate quantification of the features in the metabolomic analysis, a data normalization step is generally required. The objective of normalization is to remove undesired systematic biases so that only biologically relevant differences are present in the data. This procedure is crucial when analyzing complex biofluids like blood, where the differences in metabolic concentration between samples can be high and the introduction of internal standards is complicated. Although multiple statistical models have been developed for this objective, the two perhaps most commonly used methods are the use of endogenous stable metabolites like creatinine in urine and the use of the total spectral area. In the following slides, we will talk about the tools that can facilitate the data processing. Skyline Mass. Skyline Mass is a publicly available software developed by the University of Washington, which can support the SRM or MRM, parallel reaction monitoring, data-independent acquisition, and data-dependent acquisition with mass-1 quantitative methods and analyzing the resulting mass spectrometer data. Skyline Mass can support a quantitative analysis. Originally, this software was designed to support the proteomic analysis. You can set up the target list containing the mass information of protein fragments and apply these channels to screen the raw data for quantitative analysis. Once you align the features across different samples, you can compare the relative abundance across the sample. The bottom panel, the retention time comparison window, illustrates the alignment quality, where the samples highlighted in yellow shade may have the retention time drift. You can inspect those peaks and reintegrate the area manually. Skyline can also support the MassOne full-scan filtering, following the same principle as described in the previous slides. Skyline can also support the data-dependent acquisition or DDA search for MassOne filtering. So what is the data-dependent acquisition? In mass spectrometry, data-dependent acquisition refers to a mode of operation in which the mass spectrometer selectively collects and analyzes fragment ions from a set of precursor ions based on the intensity or quality of the signals. This type of acquisition allows the mass spectrometer to focus on the most abundant or interesting ions in the sample and reduce the amount of data that must be processed. DDA is commonly used in protein and peptide identification, where the mass spectrometer collects and identifies fragment ions derived from peptides in the sample, and then uses this information to identify the precursor peptides and the proteins they come from. The choice of precursor ions or to fragment and the choice of fragment ions to analyze is determined by the mass spectrometer software based on the quality and intensity of the signal. Parallel reaction monitoring (PRM) is a targeted mass spectrometry-based approach for the quantitative analysis of specific proteins or peptides in complex biological samples. Unlike the traditional DDA, where the mass spectrometer collects and analyzes fragment ions from a set of precursor ions based on their intensity or quality. Parallel reaction monitoring allows for the selective and specific measurement of predefined peptides regardless of their abundance in the sample. PRM involves the simultaneous monitoring of multiple peptides in a sample, using the multiple reaction monitoring transitions to quantify each peptide. The method sorts by selecting target peptides, which are then fragmented and monitored using specific mass-to-charge ratio transitions. These selective and specific measurements result in improved accuracy and precision compared to DDA and enables the quantification of low abundance peptide in complex biological matrices. Skyline can also support the data independent acquisition, which is a mass spectrometry-based approach for the analysis of complex biological samples, where the entire mass spectrum is collected and analyzed rather than focusing on specific target ions as in data-dependent acquisition or parallel reaction monitoring. In DIA, the mass spectrometer collects a full mass spectrum for each scan, and the resulting data is used to identify and quantify peptides and proteins in the sample. DIA does not rely on pre-specified targets, and instead, the entire mass spectrum is used to identify and quantify peptides, allowing for a more comprehensive analysis of complex samples. Sequential window acquisition of all theoretical fragment ions (SWAT) is one of the commonly used DIA methods, where the mass spectrometer collects full mass spectra in a sequential overlapping window manner, allowing for the analysis of all fragment ions in the samples. The resulting data is used to identify and quantify peptides and proteins in the sample, regardless of their abundance or specificity. SWAT is widely used in proteomics and has been applied to a variety of applications, including biomarker discovery, drug target identification, and clinical diagnosis. In addition, Skyline can also be used in the small molecule analysis and high-resolution metabolomics. And other applications such as collision energy optimization, ion mobility spectrum filtering, retention time prediction, and peak selection, etc. On the right is an example of using Skyline mass to perform ion mobility spectrum filtering. After applying the adequate preprocessing methods, metabolomics data is usually reduced to a feature quantification matrix. In this data representation, rows corresponds to the samples and columns corresponds to the different metabolomic features. Each metabolomic feature is intrinsically related to the concentration of a particular metabolite. Depending on the analytical technique and the spectral processing workflow that have been used, different metabolomic features are used as input for data analysis. Spectral peak area is one of the most commonly used features in high-throughput metabolomic data. They are computed through the integrations of the peaks identified and aligned as previously mentioned. Once this data has been analyzed, the identification of the metabolites representing the relevant peaks is required in order to provide biological meaning to the results. In contrast to the previous features, metabolite identification can be performed prior to data analysis in order to obtain absolute or relative metabolite concentration to be used as input for data analysis. This type of features allows both to reduce the high redundancy of peak areas, where one metabolite is often represented by multiple spectral peaks, and to provide the biological significance to all the analyzed features. In addition to peak areas and metabolite concentrations, spectral beams are also commonly used features in NMR-based studies. These techniques consist of dividing the spectra into evenly spaced regions that are later integrated to obtain the corresponding spectral beam areas. Univariate methods analyze metabolomic features independently. They are common statistical analysis approaches, and therefore, their main advantage is their ease of use and interpretation. However, the main disadvantage is that they do not take into account the presence of interactions between the different metabolic features. The metabolomic data obtained from biological samples is often very complex, with the presence of correlation between features from the same metabolite and correlation between metabolites from the same pathway. Also, the effect of potential confounding variables like gender, diet, or body mass index is not taken into account by these analysis methods, increasing the probability of obtaining false positive or false negative results. Several univariate analysis methods are available for metabolomic data analysis. The selection of the methods will depend on the statistical properties of the feature distribution. For example, when assessing differences between two or more groups, parametric tests such as the student t-test and ANOVA are commonly applied, provided that normality assumptions are conveniently verified. The latter can be confirmed using the Kolmogorov-Simonov normality test or Bartlett homogeneity of variance test. In those cases, that the normality of the data cannot be assumed. Non-parametric tests such as the Mann-Whitney U-test or Kurzweil-Wallace one-way analysis of variance are preferable. In most metabolomic studies, a large number of metabolomic features are analyzed simultaneously, and therefore, the probability of finding a statistically significant result by chance or a false positive is high. In order to control for this multiple testing issue, several correction methods are available. Each method is characterized by the particular balance between avoiding false metabolite association and prevents discarding true association. Depending on the study design, researchers might decide to use a more or less conservative approach. The Bonferroni correction is the most conservative multiple test correction approach, where the number of type 1 errors regarding to the total number of hypotheses tested as defined as family-wise error rate FWER is minimized at the expense of increasing type 2 errors. In the Bonferroni correction, the significance level for one hypothesis is divided by the number of hypotheses tested simultaneously. Although a very conservative approach, especially when the hypotheses tested are not independent, many researchers advocate using a Bonferroni correction in metabolomic studies. Other less conservative multiple test correction methods are, however, available and are mostly based on the minimization of the false discovery rate. While Bonferroni and other FWER-based methods minimize the probability of at least one false positive in the overall set of tests, FDR-based methods minimize the expected proportion of false positives on the total number of positives. Most of these methods have been extensively used for the analysis of gene expression microarray data, where thousands of genes are tested in parallel. In untargeted metabolomic studies, where large numbers of metabolites are simultaneously analyzed, and where it is also expected that more than one or two of these biomarkers will be associated, the use of less strict multiple correction methods as FDR methods might be more useful. In contrast to univariate methods, multivariate analysis methods takes into account all the metabolomic features simultaneously and consequently they can identify relationship patterns between them. These pattern recognition methods can be classified into two groups: supervised and unsupervised method. In unsupervised analysis methods, the similarity patterns within the data are identified without taking into account the type or class of the study samples. In supervised method, the sample labels are used in order to identify those features or feature combinations that are more associated with a phenotype of interest. Supervised methods are used to identify metabolic patterns that are correlated with the phenotypic variable of interest while downweighting the other sources of variance. These methods are also the basis for building classifiers based on the metabolomic features. Partial least squares is one of the most widely used supervised methods in metabolomics. It can be used either as a regression analysis, like a quantitative variable of interest, or as a binary classifier. In this case, it's PLSDA, a binary variable of interest. Unlike principal component analysis (PCA), PLS components do not maximize the explained dataset variance, but the covariance between the variable of interest and the metabolomic data. Therefore, the feature coefficients or loadings of PLS components represents a measure of how much a feature contributes to the discrimination of the different sample groups. However, one weakness of PLS is that some metabolic features that are not correlated with the variable of interest can influence the results. In order to deal with this problem, orthogonal PLS were developed. Orthogonal PLS model evolves from PLS models and factorize the data variance into two components: a first component which is correlated with the variable of interest, and a second uncorrelated component. Classification of metabolomic samples is commonly performed by fitting the discriminant analysis versions of PLAs and OPLS models. The performance of PLS and OPLS models has been extensively compared, but to date there is no agreement as to which of the two methods is superior. Support vector machines (SVMs) are another class of supervised analysis methods to build classifier based on metabolomic data. Although classifiers based on SAVM are harder to interpret, they are able to manage the presence of nonlinear relations between the metabolomic data and the variable of interest. Unsupervised methods are often applied to summarize the complex metabolomic data. They provide an effective way to detect data patterns that are correlated with experimental and/or biological variables. Principal component analysis is the most commonly used unsupervised method in metabolomic studies. PCA is based on the linear transformations of the metabolic features into a set of linearly uncorrelated variables known as principal components. These decomposition methods maximize the variance explained by the first components, while the subsequent components explain increasingly reduced amounts of variance. At the same time, PCA minimizes the covariance between these components. They are independent of each other. After applying the PCA method, a set of loading vectors and score vectors are obtained. The loading vectors represent the principal components, and each vector coefficient corresponding to the individual contribution of each variable to the principal component. The score vectors represent the projection of each sample onto a new orthogonal basis. Plotting these sample scores over the first principal components is a convenient way of summarizing the global dataset, since normally these first principal components capture most of the variability in the dataset. PCA is also used in metabolomics study to assess data quality, since it can identify sample outliers or reveal hidden biases in the study. For example, PCA has been used in several studies to determine the impact of technical variation in the analysis of metabolic profiles. Other unsupervised methods like hierarchical clustering analysis and self-organizing maps have also been applied to metabolomic data. These methods can be particularly suitable to detect nonlinear trends in the data and are not conveniently covered by PCA. SOM have been used in metabolomic studies to visualize the metabolic phenotypes and feature patterns, as well as to prioritize the metabolites of interest based on their similarity. HCA is also a powerful clustering and visualization tool that provides a clustering procedure at the feature and sample levels according to a predefined distance measure. Metabolite identification is one of the major challenges of high-throughput metabolomic analysis. In mass-based studies, the common metabolite identification approach is based on querying metabolomics databases for the neutral molecular mass values of the identified peaks using a tolerance window. The neutral molecular mass is inferred from the peak mass-to-charge ratio and depends on the chemical nature of the identified peak. Assuming no prior knowledge, each peak mass-to-charge ratio can lead to multiple plausible neutral molecular masses that can represent different ionization adas. This multiplicity often results in a high number of false positive identification. In order to reduce false positive, several methods have been developed. Upstream and Camarade are methods designed to identify isotopic and ADAT patterns in order to reduce data complexity in mass experiments. Using this approach, the chemical nature of each selected ion peak is estimated, and only one neutral mass is inferred from each identified pattern. Using these feature reduction methods has the added advantage of improving the ascertainment of true biological compounds. Metabolite spectral databases are essential for metabolite identification. The quality of the stored data, as well as the number of metabolite spectra available in these databases, is critical for the performance of identification algorithms. These slides summarize the commonly used mass spectral database for your reference. We will go through them in the following slides. The NIST, National Institute of Standards and Technology Mass Spectral Database, is a comprehensive collection of mass spectral data that is used to identify and characterize chemicals and chemical compounds. The database contains about 500,000 spectra, including data on small molecules, biological and environmental compounds, and pure substances, as well as information on fragmentation patterns and spectra from various mass spectrometry techniques. The NIST mass spectral database is widely used by researchers, industry, chemists, and government agencies for a variety of purposes, including chemical analysis, quality control, and environmental monitoring. It is recognized for its high quality, accuracy, and reliability, and is considered a valuable source for scientists and technicians in the field of mass spectrometry. The associated tool, Mass Interpreter, can find possible structural origins of peaks in the mass spectrum and provides formulas and isotopic processing utilities. MassBank is an open source mass spectral library that is maintained and developed in Europe. MassBank is a publicly accessible database that contains mass spectral information and related data for small organic molecules. It was created as a resource for scientific researchers, particularly those in the field of metabolomics and environmental analysis. MassBank contains spectra for thousands of compounds, including drugs, metabolites, and environmental pollutants, as well as related information such as a molecule formula, molecule weight, and chemical structure. The database is widely used for the identification of compounds in complex mixtures and for the development of new mass spectrometry-based analytical techniques. MZCloud is also a publicly accessible online database of mass spectrometry data and related information. However, only IE browser is supporting the operation of MZCloud. The Human Metabolome Database (HMDB) is perhaps the most extensive public metabolomic spectral database today. The Human Metabolome Database is a freely available electronic database containing detailed information about small molecule metabolites found in the human body. It is intended to be used for applications in metabolomics, clinical chemistry, biomarker discovery, and general education. The database is designed to contain or link three kinds of data: chemical data, clinical data, and biochemistry data. The database contains 220,945 metabolite entries, including both water-soluble and lipid-soluble metabolites. Additionally, 8,610 protein sequences are linked to these metabolite entries. Each metabol card entry contains 130 data fields, with two-thirds of the information being devoted to chemical and clinical data, and the other one-third devoted to enzymatic or biochemical data. Many data fields are hyperlinked to other databases and a variety of structure and pathway viewing applets. Medline also has a straightforward user interface. You can just enter the accurate mass value and specify the ionization mode and ADA forms you are looking for. Then Medline will return a list of candidates or possible hits based on your search. Lipidomic database collects a number of different lipid categories, from fatty acid, glycerolipids to polyketides, etc. If you aim to perform a lipidomic analysis, you can use this database as a reference library. FoodDB is a database of food-related information, including information on the composition and nutritional value of different foods. FoodDB associates the nutrients and ingredients in the natural food. If you want to perform a food-based suspect screening and try to probe the primary markers for food authentication, then this may be the place you'd like to check it out. Aside from the publicly available databases, you can also gather relevant information and formulate your own suspect screening list. This kind of personalized compound database and library requires at least the following elements: molecule formula, accurate mass, compound name, and if any, the collision cross-section value. We talk about the metabolite identification and library matching, but how can we communicate the confidence in suspect screening? Szymanski et al. Provides a structured framework to communicate the confidence level in suspect screening from level 5 to level 1 with increasing confidence in compound identification. Level 1 is the confirmed structure, representing the ideal situation where the proposed structure has been confirmed via appropriate measurement of a reference standard with mass, and retention time matching. Level 2 is the probable structure, indicating that it was possible to propose an exact structure using different evidence. The sub-level 2A involves matching the literature or library spectrum data where the spectrum structure match is unambiguous. Care is needed when comparing spectrum recorded with different acquisition parameters like resolution, collision energy, ionization, or mass level to ensure the validity of the match, and decision criteria should be clearly presented. Desirable additional evidence such as a retention behavior would require, for example, a retention index for both the measured and matched spectrum established for GC-MAS but not yet sufficiently for LC-MAS based techniques. The sub-level 2B represents the case where no other structure fits the experimental information, but no standard or literature information is available for confirmation. Evidence can include diagnostic mass fragments and ionization behavior, parents compounds information, and experimental context. Although the breakdown into A and B is useful for research purpose, for practical reporting, a level 2 classification may often suffice as probable structure indicates the confidence in the candidate. Tentative candidate describes a gray zone where evidence exists for possible structure but insufficient information for one exact structure only. Although there are many shades of gray possible for many different situations, for all intents and purposes, the exact structure remains speculative at level 3. Example includes the position of the hydroxylation was not unequivocal as shown in the yellow shade. Further examples are top-ranked structures from in silico fragmentation of candidates from compound databases searches, and suspects selected using additional information such as high predicted likeliness of being a transformation product, the number of references, or retention behavior. Level 4 unequivocal molecular formula is possible when a formula can be unambiguously assigned using the spectral information, but insufficient evidence exists to propose possible structure. The mass could be uninformative, contain interferences or not even exist. However, the formula provides some information and is worth presenting as it can be traced in future studies. Level 5, exact mass, can be measured in the sample and be of specific interest for the investigation, but lack information to assign even a formula. Screening and non-target methods allows the tracing of these masses in other investigations, but the level 5 indicates that no unequivocal information about the structure or formula exists. It is even possible to record the mass of a level 5 mass and save it as an unknown spectrum in the database. This level should only apply to a few masses of specific interest, since it would be counterproductive to label all masses in a sample as level 5. Blank measurements should be used to ensure the substance do not arise from sample preparation or measurements. Lastly, we will introduce the common metabolomics approaches used for biological interpretation, including pathway analysis and correlation-based network analysis. Pathway and network analysis approach increase the information generated by metabolomic studies. Both approaches exploit the relational properties present in the metabolomic data. Pathway analysis uses a prior biological knowledge to analyze metabolite patterns from an integrative point of view. Alternatively, network analysis uses the high degree of correlation existing in metabolomic data to build the metabolic networks that characterize the complex relationship existing in a set of major metabolites. Metabolic pathways are groups of metabolites that are related to the same biological process and that are directly or indirectly connected by one or multiple enzymatic reactions. Biological databases such as Kyoto Encyclopedias of Genes and Genomes (KEGG), Small Molecule Pathway Database (EHMN), Wiki Pathways, and MetaPsych provides exhaustive information of a large number of metabolic pathways. The availability of this data is therefore enabling the use of pathway-based approaches in metabolomics. These methods are currently referred to as metabolite set enrichment analysis MSEA, and are methodologically based on the gene set enrichment analysis approach designed for pathway analysis of gene expression data. To date, three different approaches have been developed to perform MSEA, including the overrepresentation analysis (ORA), quantitative enrichment analysis (QEA), and single sample profiling (SSP). In overrepresentation analysis, a hypergeometry test or a Fisher's exact test is used to evaluate whether the metabolites of these groups are represented more than expected by chance. When the input metabolite list is defined as the set of metabolites which are differentially expressed in the analyzed phenotypes, the ORA resource may identify metabolic pathways that are globally associated to these phenotypes. Unlike ORA, in quantitative enrichment analysis, the input data is a set of metabolite concentrations from multiple samples. Enriched pathways can be identified using different approaches like a global test, global ANCOVA, or the weak causing-based test. Enriched pathways include pathways where a few number of compounds are significantly changed, or pathways where a large number of metabolites are slightly but consistently changed. While the two previous methods are suited for studies involving large number of samples. Single sample profiling can be used at the sample level. The input data for SSP analysis is an input list of normalized metabolite concentration in a common biofluid, tissue, or cell type, and a database with the normal concentration range of these metabolites in the sample. From these input data, SSP identifies the set of metabolites showing levels significantly different from the normal concentration range. These are the MSEA tools for your reference. One of the main features of biologic data is the high level of correlation existing between the different elements. Part of this relational patterns is due to the metabolites that belong to a common metabolic pathway. In other cases, however, the observed correlation may be due to other causes like global perturbations, specific perturbations, and or the intrinsic variability of metabolomic data. Consequently, metabolites that do not show significant differences across the study phenotypes may still show different correlation patterns with other metabolites in each phenotype. These patterns can provide valuable information about the underlying metabolic network associated to a specific biological process. Unlike pathway analysis, the correlation-based methods build a metabolite network according to the relationship patterns observed in the experiment data. In the resulting network, each metabolite is represented by a network knot. But in contrast to pathway analysis, the link between knots represents the level of mathematical correlation between each pair of metabolites. In metabolomics data, high correlation coefficients are frequent due to the presence of systematic and indirect association. Using classical correlation coefficients leads to highly crowded networks where direct and indirect association are not distinguished. This problem can be successfully overcome using partial correlation. In this approach, the correlation between two metabolites is conditioned against the correlation with the remaining metabolites. Consequently, partial correlation allows to discriminate between the direct and indirect metabolite correlations. Metaboanalysis is a widely used metabolomic data analysis tool. In the following slides, you will learn the basic structure of this tool for your own metabolomic studies. Depending on the different types of input data, Metaboanalysis supports different input files from the mass spectral raw data to organize a concentration table, time series data, etc. The platform itself also provides the data templates for your reference. Metaboanalysis can fulfill a variety of different MSEA analyses. In the following slides, we will go through each text individually. Metaboanalysis supports the generation of a heat map, so we can visually compare the metabolite's concentration and abundance across the samples. Metaboanalysis also supports the pathway analysis in a simple, clear interface. The pathway analysis results, the disturbed KEGG pathways, and the affected metabolites in those pathways will be shown. A link to the KEGG database of a specific pathway will also be provided. Metaboanalysis can also perform the correlation-based network analysis. The network interactions of genes or metabolites will be visualized. The shorter the distance between the two metabolites, the stronger the correlation they are. That's for today. Enjoy.